Hello, everybody. In this lecture, we're going to talk about cell division and transport into the cell, specifically for eukaryotes. So let's get right into it. Now, by the end of this module, you should be able to differentiate between mitosis and meiosis, and differentiate between phagocytosis, pinocytosis, and receptor-mediated endocytosis. And finally, you should be able to define exocytosis. And so in cell division in eukaryotes, you can actually have a couple of different options. One, we have to think about their reproduction. So in bacteria, all reproduction is asexual. However, in eukaryotes, you can have sexual and or asexual reproduction. Regardless, before that cell is able to divide, it's going to have to copy its genetic material. And just in comparison, cell division is longer in eukaryotes because not only is their genome larger, they've got more genes, more base pairs, they also have to replicate all those intracellular structures that they have, all those organelles, which the prokaryotes are lacking. And so in mitosis, you get basically the generation of two identical offspring. This is similar to binary fission in the sense that you go from a single cell to two daughter cells. However, it's a much more involved process than binary fission is. Now, in this case, the offspring cells are going to have the same number of chromosomes as the parent cell. And it's going to be the process is going to be divided into, we've got the interphase, the prophase, late prophase, then we go into metaphase, anaphase, telophase, and then cytokinesis, where they break apart into two distinct cells. Now, this is not a cell biology class, and I'm not going to make you remember that all the things that are happening in each of those phases, although if you're a biology major, or if you're interested in that in the biological aspect, I highly suggest that you do know those things. What I want you to really know here is these two bits of information that we've got there on the slide. Now, meiosis is a little bit more involved than mitosis. This is going to be involved in sexual reproduction. It has two separate cell division stages. And so in this case, your one parent cell is going to produce 4 gametes. And so gametes are the sexual reproduction cells. These are haploid, which is different from the normal cell, which is diploid, which is like single copy of the chromosome versus 2 copies of the chromosome. And in this process, there's an occurrence of crossing over, which does allow for genetic recombination. This is important for genetic diversity in eukaryotes. But what you see here is you have your prophase 1, metaphase 1, anaphase 1, telophase 1, and then prophase 2, metaphase 2, anaphase 2, and telophase 2. And at the end of telophase 2, you've got cytokinesis, which gives you 4 cells, 4 daughter gametes that are haploid. So that's the important part. Again, knowing the exact specific things that are happening at these stages, I'm not super worried. I do want you to know the difference between mitosis and meiosis in terms of what they're producing. But you don't have to know everything that's going on in each of these phases. And so here's a little, explanation of what's happening in these different types of division that we've talked about. We've got our mitosis, meiosis, and binary fission. Mitosis and meiosis are eukaryotic processes, while binary fission is our prokaryotic process. And it also happens in mitochondria and chloroplasts, or at least something that's very similar to binary fission. When in mitosis, you need to know that you have your diploid daughter cells, and in meiosis, you need to know that you have your haploid daughter cells, which are gametes, which are used in sexual reproduction. All right, so we're going to talk a little bit about cellular transport in eukaryotes, endocytosis and exocytosis. So again, remember, biochemists, they love to define things for what it does. So endo means import or into, and cytosis would be like the cell. So if you import into the cell, you're endocytosing. And if you're exporting things out of the cell, that's exocytosing or exocytosis. So in endocytosis, what you're trying to do is get something into the cell, right? And so what's going to happen is the plasma membrane is going to fold around whatever it is that you have in that outside of the cell, and then it's going to pinch off the membrane. So your two edges that pinch around it are going to pinch off into its own little vesicle. And this is like a little, what we call an endocytic vesicle, and that's going to contain your substance. Now that's going to come in a couple of different forms. We got phagocytosis, pinocytosis, and receptor-mediated endocytosis as big groups of things that we want to talk about. And pinocytosis, which is essentially translates to cell drinking. This is endocytosis of dissolved substances or in these really small vesicles. And so you see here, this little square represents some sort of ion or something that's been dissolved. You get your pseudo like membrane that looks kind of like a pseudopod, right? And it folds in, it pinches off, and then you get this vesicle that's containing your dissolved product. Now, phagocytosis translates to cell eating. This is going to be endocytosis of undissolved substances. So pinocytosis is the dissolved stuff, so they're just drinking. Endocytosis is the undissolved stuff, so they're eating. Now, I've got a little video here of a macrophage endocytosing blood cells from a different animal. So basically, all these little guys are the blood cells, and this big thing in the middle is your macrophage. And so this is a form of phagocytosis. So I'm going to go ahead and play this video. And so phagocytes in cells are these specialized immune system cells, and this is a macrophage. And what's going to happen is the cell is going to engulf its target. And we see that happening here. These red blood cells are foreign cells, and they are not supposed to be in the body. So these are the blood cells of a sheep, but these are macrophages from not a sheep. And so when the cell engulfs its target, it forms a phagosome. That phagosome is going to fuse with what's called a lysosome. And the lysosome contains these hydrolytic enzymes. Those hydrolytic enzymes are able to chew up the whatever gets phagocytosed. And then when those two things fuse together, those lysozymes get access to your phagocytized cell or product, in this case, these blood cells. And in most cases, these hydrolytic enzymes are going to kill bacteria, they're going to kill viruses. Occasionally, those things have resistance. Ultimately, the waste products that are going to be produced are going to get expelled, probably through exocytosis, actually. All right. And so here's a little like diagram of phagocytosis. We see that the target is here. You get this phagosome, which is like it's got your pinched off plasma membranes surrounding that target. And then it's going to fuse with this lysosome, which has these hydrolytic enzymes. The hydrolytic enzymes are going to come in and break up whatever that target is. Now you're going to have all this waste that's going to be produced, and that's going to get exocytosed from the cell, right? And over here, we have an image of Staph aureus actually getting phagocytosed by a neutrophil. Now we have receptor-mediated endocytosis. This is, I got a bad picture here. Let's see if I got a better one. Now, sorry about this. You've got these ligands, which you can kind of see here. It's like, let's think of this star as being a ligand. Over here, we have our receptor. It's this little green thing that's sticking off the edge. And so these ligands can be hormones or nutrients. They can be pathogens, but they're going to bind to something specific. And so what they're going to do is they're going to bind to these receptors. The inner surface of the plasma membrane is going to be coated with clathrin. And the clathrin is going to form a pit. We'll see it in this next picture a little bit better. And then you're going to get this pinched off clathrin coated pit, which is going to form a vesicle. That's going to form with an endosome. That endosome is going to alter its pH, which is going to separate your ligand and receptor, and then those things are going to get delivered where they need to be. And so if we look here, this is a better representation. You can see the whole thing. We've got our ligand attaching to our receptor here on the cell surface. That ligand is going to be specific to that receptor, so it's not going to be just any old thing. Now, when you get that, you're going to have this clathrin that's going to coat here. You get this formation of a pit. It's going to pinch off. You're going to get fully clathrin coated vesicles. They're going to ultimately release and then bind with this endosome, which is acidic. Now, when they're bound, that alteration in pH is going to change the actual structure of the receptor, and that receptor is then going to release the ligand. And now that they're released, they can be separated, and that ligand, that receptor can be released back to, through exocytosis, back to the cell surface and recycled. And then your target can then bind with a lysosome. So now we're going back into the phagocytosis pathway, and the ligand can then get broken down by your hydrolytic enzymes. And so if they need to be released into the extracellular matrix, then they can be exocytosed, or if they need to be processed internally in the cell, they can be released inside the cell. And so we've talked a little bit about this overall. Exocytosis is the process of basically taking things out of the cell. And so that happens both in these process we talked about where you're getting rid of waste, but also when you're secreting cargo that was produced in the cell, such as proteins that are going to be released extracellularly, or lipids and stuff like that. And so what's going to happen is you're going to get the formation of these vesicles at your Golgi apparatus. They're going to be transported along the cellular highways, right, through your along your cytoskeleton, and they're going to end up outside of the cell where they can excrete it. And this can be anything from your, like I said, these proteins and lipids that are made inside the cell, or it can be waste products. It can be toxic buildup of products that the cell can't have any more of. And this is the same thing that happens with neurotransmitters. So when you're thinking about the way that neurons work, this is also through a process of exocytosis. All right, so you should now be able to differentiate between mitosis and meiosis, and between these three types of endocytosis, as well as define exocytosis. All righty, I hope you enjoyed, and I'll see you soon.